In this video, I will be explaining the BSc 5th semester subject Zoology, Immunology and Animal Biotechnology as prescribed by Osmania University and other universities of Telangana. We will go through the important questions and answers one by one. Let's begin with a quick revision. Unit 3 first important short question is Plasmids. Plasmids are small, circular DNA molecules found in bacteria that replicate independently of the bacterial chromosome. They are not essential for survival but often carry useful genes, like antibiotic resistance. Key features: circular and double-stranded DNA. Can replicate on their own. Carry genes that can provide an advantage, example, resistance to drugs or production of toxins. Importance in biotechnology. Plasmids are widely used as cloning vectors to insert foreign genes into bacteria. Scientists can cut plasmids with restriction enzymes, insert a gene, and then let bacteria produce the protein encoded by that gene. Example: PBR322 plasmid is a common plasmid used in labs for gene cloning. Plasmids have been used to produce insulin, growth hormone, and vaccines. Fun way to remember: plasmid is equal to tiny DNA ring that can carry cool genes into bacteria like a delivery van. Next important short question is 2 cosmids. Cosmids are hybrid cloning vectors that combine features of plasmids and bacteriophages, viruses that infect bacteria. They are designed to carry larger DNA fragments than plasmids, making them very useful in gene cloning. Key features: circular DNA like plasmids, but packaged in a virus coat for easy delivery into bacteria. Can carry up to 40 to 45 kilobyte of DNA, much bigger than plasmids. Contain origin of replication from plasmid and cause sites from phage for packaging into viral particles. Importance in biotechnology. Cosmids are used to clone large genes or gene clusters that plasmids cannot handle. After entering bacteria, the DNA behaves like a plasmid and replicates independently. Example: used in genome projects to clone human DNA fragments. Fun way to remember: cosmid is equal to a plasmid wearing a virus coat, carries big DNA packages into bacteria. Next important short question is 3 shuttle vectors. Shuttle vectors are special cloning vectors that can replicate into different species, usually bacteria and yeast or bacteria and mammalian cells. This makes them very useful for transferring genes between organisms. Key features: contain 2 origins of replication, one for each host. Carry selectable markers to identify cells that have taken up the vector. Can carry foreign genes for expression in multiple hosts. Importance in biotechnology. Shuttle vectors are used when scientists want to clone a gene in bacteria, fast growth, and then study or express it in eukaryotic cells. They save time and make genetic experiments more flexible. Example: a gene cloned in a bacterial shuttle vector can later be expressed in yeast or mammalian cells to produce a protein. Fun way to remember: shuttle vector is equal to a DNA bus, carries genes between 2 different hosts safely. Next important short question is 4 restriction endonucleases. Restriction endonucleases. Restriction endonucleases, also called restriction enzymes, are special proteins that cut DNA at specific sequences. They act like molecular scissors, allowing scientists to cut and manipulate DNA precisely. Key features: recognize specific DNA sequences, usually 4 to 8 base pairs. Cut DNA either in staggered ends, sticky ends, or blunt ends. Found naturally in bacteria as a defense against viruses. Importance in biotechnology. Restriction enzymes are essential tools in recombinant DNA technology. They allow scientists to cut plasmids and foreign DNA at specific spots so genes can be inserted, cloned, or studied. Example: E. coli cuts DNA at the sequence gut, producing sticky ends that can join with other DNA fragments. Used in gene cloning and creating genetically modified organisms. Fun way to remember. Restriction enzymes is equal to DNA scissors, cut at the right spot, and you can paste anything you want. Next important short question is 5. Transgenic sheep. Transgenic sheep. A transgenic sheep is an animal that has foreign genes inserted into its DNA. These genes can help produce useful proteins or study human diseases. How they are made. 1. A desired gene is inserted into a fertilized sheep egg using techniques like microinjection. 2. The egg is implanted into a surrogate mother sheep. 3. The offspring carry the foreign gene in all their cells, making them transgenic. Importance. Transgenic sheep can produce therapeutic proteins in milk, such as human antithrombin, prevents blood clots. They are used in research to study genetic diseases and improve livestock traits. Example: ATRYN sheep produce antithrombin in their milk, used as medicine for patients with blood clotting disorders. Fun way to remember. Transgenic sheep is equal to little genetic factories, sheep making human proteins in milk. Next unit 3. Long questions. First important long question is 1. Elaborate on concept and scope of animal biotechnology. Concept and scope of animal biotechnology. Introduction. Animal biotechnology is a branch of science that uses modern biological techniques to study and improve animals and their products. It combines genetics, molecular biology, and reproductive technologies to produce animals with desirable traits, enhance productivity, and develop medicines. Concept. It involves manipulating genes and cells to improve livestock, fish, or other animals. Techniques include recombinant DNA technology, cloning, transgenesis, and cell culture. Focus is on healthier animals, higher yields, and disease resistance. Scope and applications. 1. Agriculture and livestock improvement. Producing animals with better meat, milk, or wool quality. Example: transgenic cows producing more milk or sheep producing therapeutic proteins. 2. Medical applications. Animals can act as bioreactors to produce medicines like insulin, growth hormones, and clotting factors. Used in research for human diseases, drug testing, and vaccine development. 3. Conservation biology. Helps preserve endangered species through cloning and cryopreservation of gametes or embryos. 4. Industrial applications. Producing enzymes or proteins in animals that can be used in food, pharmaceutical, or chemical industries. 5. Research and biotechnology development. Provides models for genetic studies, understanding disease mechanisms, and developing new treatments. Example: ATRYN sheep producing antithrombin in milk. Cloned animals like Dolly the sheep helped understand mammalian cloning. Fun way to remember. Animal biotechnology is equal to mix of genes, cells, and tech to make animals healthier, stronger, and more useful. Conclusion. Animal biotechnology is revolutionizing agriculture, medicine, and research. Its scope is vast, from improving livestock productivity to producing human medicines and saving endangered species. Understanding this field is essential for future scientists, veterinarians, and biotech researchers. Next important long question is to explain recombinant DNA technology and its applications. Recombinant DNA technology, rDNA, and its applications. Introduction. Recombinant DNA technology is a method of joining DNA from different sources to create new genetic combinations. It allows scientists to manipulate genes and produce useful proteins, medicines, and genetically modified organisms, GMOs. Basic steps of rDNA technology. 1. Isolation of gene. The desired gene is extracted from a donor organism. 2. Cutting DNA. Both the gene and vector DNA, plasmid, cosmid, or shuttle vector, are cut using restriction endonucleases. 3. Ligation. The gene is joined into the vector using DNA ligase, forming recombinant DNA. 4. Introduction into host. The recombinant DNA is inserted into a host organism like bacteria, yeast, or animal cells. 5. Selection and screening. Only the cells with recombinant DNA are selected using markers. 6. Expression. The host expresses the gene, producing the desired protein or trait. Applications of rDNA technology. 1. Medical applications. Production of human insulin, growth hormones, clotting factors, vaccines. Gene therapy for treating genetic disorders. 2. Agriculture and animal biotechnology. Creation of transgenic animals with better traits. Genetically modified plants and animals resistant to diseases or environmental stress. 3. Industrial applications. Microbes producing enzymes, biofuels, and biodegradable plastics. 4. Research. Helps in studying genes, proteins, and disease mechanisms. Example. Human insulin produced by inserting the human insulin gene into E. coli, which then produces insulin in large quantities. ATRY in sheep producing antithrombin in milk is another example. Fun way to remember. rDNA is equal to cut, taste, and play with DNA, make cells produce whatever protein we want. Conclusion. Recombinant DNA technology is a powerful tool in biotechnology. It revolutionized medicine, agriculture, industry, and research, making it one of the most important technologies of the 21st century. Next important long question is 3. Define cloning and explain the process of cell cloning. Cloning and the process of cell cloning. Introduction. Cloning is the process of creating genetically identical copies of an organism, cell, or gene. In cell cloning, a single cell is used to produce a population of identical cells. This is important in research, medicine, and biotechnology. Definition. Cloning, making a copy that is exactly like the original in DNA. Cell cloning, producing identical cells from a single parent cell in a controlled environment. Process of cell cloning. 1. Isolation of a single cell. A healthy cell is selected from the organism or tissue. 2. Culturing the cell. The cell is placed in a sterile nutrient medium containing growth factors. Under suitable conditions, the cell divides repeatedly. 3. Formation of a clone. The single cell multiplies into a colony of identical cells, all carrying the same genetic information. 4. Selection and expansion. Specific cells with desired traits can be isolated and grown further for research or applications. Applications of cell cloning. Gene cloning, producing multiple copies of a gene for research or medicine. Medical research, studying disease mechanisms or testing drugs. Animal biotechnology, producing transgenic animals using cloned cells. Example. Cloning of insulin-producing cells for diabetes research. Dolly the sheep, though an example of whole animal cloning, started from a single somatic cell. Fun way to remember. Cell cloning is equal to one cell starts a family of identical twins in a dish. Conclusion. Cell cloning is a fundamental technique in biotechnology. It allows scientists to study genes, produce therapeutic proteins, and develop transgenic animals. Understanding cell cloning is key to modern research and medical advances. Next important long question is: 4. Explain about the animal cloning methods in detail. Animal cloning methods. Introduction. Animal cloning is the process of producing genetically identical animals. It is a key tool in animal biotechnology for research, agriculture, and medicine. There are several methods, each with its own approach. 1. Embryo splitting, blastomere transfer. Early-stage embryos are split into individual cells. Each cell develops into a complete embryo. The embryos are then implanted into surrogate mothers. Example, used in livestock breeding to produce identical twins in cattle or sheep. Advantage: simple and natural. 2. Somatic cell nuclear transfer, SCNT. A mature somatic cell, body cell, is taken from an animal. Its nucleus is transferred into an enucleated egg cell without nucleus. The egg develops into an embryo with DNA identical to the donor. Example, Dolly the sheep was produced using SCNT. Advantage: can clone adult animals. 3. Induced pluripotent stem cells, iPSCs. Somatic cells are reprogrammed to become pluripotent stem cells. These cells can develop into any tissue or complete organism under lab conditions. Mostly used in research rather than commercial cloning. Applications of animal cloning. Agriculture, producing high-yield livestock. Medicine, creating transgenic animals for drug production or disease studies. Conservation, helping endangered species survive. Fun way to remember. Animal cloning is equal to embryo splitting for twins, SCNT for adult copies, stem cells for lab miracles. Conclusion. Animal cloning methods allow scientists to replicate animals with desirable traits, study diseases, and preserve biodiversity. Among them, SCNT is the most famous for producing clones from adult animals, while embryo splitting is simpler for young embryos. Understanding these methods is essential in modern animal biotechnology. Next important long question is: 5. What is transgenesis, and explain various methods of transgenesis. Transgenesis and methods of transgenesis. Introduction. Transgenesis is the process of introducing a foreign gene, transgene, into an animal's genome so that the animal can express a new trait. Transgenic animals are widely used in research, medicine, and agriculture. Definition. Transgenic animal, an animal that carries a foreign gene stably in its DNA. Transgenesis, the process of creating such animals. Methods of transgenesis. 1. Microinjection. The foreign gene is directly injected into a fertilized egg under a microscope. The egg is implanted into a surrogate mother. Example, transgenic mice and sheep producing therapeutic proteins. Advantage: simple and widely used. 2. Retroviral method. Retroviruses are used to carry the foreign gene into early-stage embryos. The virus integrates the gene into the host genome. Example, producing transgenic chickens. Advantage: efficient for multiple copies of a gene. 3. Sperm-mediated gene transfer, SMGT. Foreign DNA is attached to sperm cells, which then fertilize eggs. The resulting embryo may carry the transgene. Example, used experimentally in livestock. 4. Embryonic stem cell method. Gene is inserted into stem cells derived from embryos. Modified stem cells are injected into another embryo, producing chimeric transgenic animals. Example, transgenic mice used in research. Applications. Medicine, animals producing human insulin, clotting factors, or antibodies. Agriculture, livestock with disease resistance, improved milk or meat. Research, studying human diseases and gene function. Fun way to remember. Transgenesis is equal to give animals a genetic upgrade, inject, infect, or hitch a ride with sperm. Conclusion. Transgenesis is a powerful tool in modern biotechnology, allowing scientists to produce new traits, study genes, and make medicines. Microinjection and retroviral methods are the most commonly used techniques, while stem cell and sperm-mediated methods provide alternative approaches. Understanding transgenesis is key to genetic engineering in animals.